just because we didn't finish the last video on translation as fast as I anticipated. It actually went a little bit long. Excuse me. So to begin this video, we're going to go with ribosomal translocation, what happens after translation. So ribosomes can either be in the cytosol or cytoplasm in the cell, and those are known as free ribosomes, which are over here. And then we have endoplasmic reticulum, or ribosomes that are located on the endoplasmic reticulum. So I'll just draw the cell real quick so that we have an example of what. So we have our cell, we have our nucleus, and then we're going to have ribosomes just chilling somewhere in the cytoplasm. Oof. This is going to be really good. So yeah, chilling in the cytoplasm, and then we'll have the ER, which is kind of close to the nucleus, right there, and it's got some membrane folds. And we're going to have ribosomes on the ER, the endoplasmic reticulum. So, goodness gracious, yeah, this is not looking good. Not an artist, scientist. All right, so the ribosomes located on the endoplasmic reticulum, once they successfully translate the mRNA and the tRNA, using the tRNA into a polypeptide. I'm going to zoom in here on this drawing. You see here that the ribosome is like linked. So on the peptide, the polypeptide that's synthesized from the mRNA, see we have methionine, which is an amino acid, lysine, and I added leucine, which is this one right here, just because I want our peptide to have some length to it, just so you know, better visualize what's happening. So the signal recognition sequence, this is something new. So the signal recognition sequence is a group of amino acids, I believe, that are coded for before the initial starter codon and methionine. This sequence is added to basically give direction to where the translated polypeptide is going to go. And something called the signal recognition particle binds to that signal recognition sequence. So that happens during translation. So once the polypeptide, the translated polypeptide, gets to the endoplasmic reticulum using the signal recognition sequence and the signal recognition particle, the signal recognition particle is going to bind, so the signal recognition particle, SRP, is going to bind to the signal recognition particle receptor. So after it binds, this transmembrane channel, which is opening to the lumen of the ER, so the endoplasmic reticulum, it's going to bind, it's going to open this channel. And because of the signal recognition particle, signal recognition sequence, and the signal recognition particle receptor, it's going to line up in such a way that allows the translated polypeptide into the lumen of the endoplasmic reticulum. So basically the sequence gets bound by a particle. The particle signals to the signal recognition particle receptor. It opens the gate, the transmembrane channel, and allows the polypeptide to enter. After the polypeptide enters, something called the signal peptidase degrades the signal recognition sequence, the amino acids at the beginning of the sequence that aren't necessary, because all we wanted is what we translated. We don't want the thing that helped us get to the ER. Um, so it's signal peptidase degrades the signal recognition sequence. And the majority of what you need to know is that the signal recognition particle binds to the signal recognition sequence, and they help facilitate the journey to the endoplasmic reticulum, which we marked up here. Um, so the ribosomes on there, they get shipped to the endoplasmic reticulum, and they get into the lumen. And the whole point of that is to protect the translated polypeptides, because sometimes the environment, the cytosolic environment, is different, and the proteins are sensitive. We know that proteins get denatured by, acidity, heat, and other conditions that aren't like best for the enzyme to work in. If you've ever did work with enzymes, you know that they function best at certain pHs and certain temperatures. So the lumen of the endoplasmic reticulum sort of serves as a safe space for these polypeptides until they're shipped to where they need to go. But that's later. So then we have free ribosomes, the ribosomes that are just outside of this nucleus and in the cytoplasm, just chilling. First, endoplasmic reticulum ribosomes, they synthesize proteins that are destined for to be secreted out of the cell, to become part of to become like proteins near the phospholipid bilayer. So there are proteins in there, so like channels and stuff. They're basically secretory proteins, and also they can become part of lysosomes, or not vacuoles, endosomes, but mostly lysosomes. So just they're destined to be integrated into a membrane. If they're integrated into a membrane, they're probably from the endoplasmic reticulum, or if they're secretory. So just remember that. The ER makes secretory proteins and membrane bound, or not membrane bound, but proteins that are destined for the membrane. There we go. Okay. So now, free ribosomes. Free ribosomes floating in the cytoplasm synthesize polypeptides that are destined to go be a part of the cytosol, so cytosolic proteins, proteins that are involved in glycolysis, so those enzymes. The first one I can think of is like phosphofructokinase 1. If you know anything about glycolysis, you're probably familiar with that. Transcriptional proteins, so nuclear enzymes. So any thing I can think of right now is like polymerases. That's the biggest one for transcription anyway, but mostly nuclear enzymes. So you'll probably have some enzymes that are involved in mitosis as well, which will be another video later down the road. Mitochondrial enzymes. Mitochondria is a powerhouse of the cell. If you're familiar with the Krebs cycle, the electron transport chain, proteins that are involved in that definitely get synthesized in the free ribosomes. And then peroxisomal enzymes, which we know are good for, beta oxidation and dealing with reactive oxygen species. But those could all be videos, to be honest. But we're just gonna keep on chugging along. If enough people express interest in an explanation of those pathways, I would be happy to go through them. Because at the end of the day, you can never go over the basics too much. I mean, Tim Duncan, Hall of Fame, hit the backboard, hit the square, man. It's the same concept for science, same concept for life, honestly. Just hit the same, master the basics, and the rest will follow. But yeah, free ribosomes, they synthesize cytosolic proteins, glycolysis, proteins that are involved in glycolysis, transcriptional proteins, nuclear enzymes, so enzymes involved in the nucleus, mitochondrial enzymes, and peroxisomal enzymes. Lastly, post-translational modifications, PTMs, if you will. No, that's bothering me. Um, so, post-translational modifications are after the polypeptide has been synthesized. It needs direction on where it wants to go. So, part of that entails marking it essentially, and so you can mark it with these different molecules, and that sort of shows um, the polypeptide where to go, when to be activated. It's a really intricate system, but it it can set it up to be activated. Um, there's countless examples. Um, so if you look up any of these terms, you'll see a large amount of applications for these various post-translational modifications. So first, glycosylation, glyco, glyco. We're gonna have a sugar. Um, so if you add a sugar or a glucose molecule to something, it's probably destined for a GLUT2 or GLUT4 transporter. Um, so if you need a protein to go into a cell, and you need that cell, um, maybe it's like a cell in the stomach, and you need to get it into the bloodstream. Um, it's going to utilize one of those GLUT or GLUT4 transporters. Uh, Potentially maybe the liver. I'm not correctly. Don't quote me on that. Too sure. I need to do more research on that. But um, glycosylation basically it adds a glucose group or sugar, and destines it for a transporter that is responsible for such. You have lipidation, which is adding like a lipid molecule for cell membranes. If you're familiar with prostaglandins or perhaps sphingolipids, these are all different types of lipids that, you know, when attached to a protein, can alter its function. These are typically destined for the cell membrane just because it's a phospholipid bilayer, and a lot of the things with molecules and proteins that have lipids are destined to join the lipid membrane. Um, Not too much to say about that. Um, there's probably a bunch you could say about it, but I just don't know it yet, unfortunately. But that's lipidation. Anyways, number three, phosphorylation. If you know what that means, you're adding a phosphate group. The negatively charged phosphate group can do a bunch of things. It's most notoriously known in protein kinase A, if you're familiar. Uh, Activates most signaling pathways. I think it's in, I want to say G protein coupled, um, G protein linked receptors. You have protein kinase A, which activates and then does a cascading sequence, but I could be wrong. Again, here to learn, teach what I know, and hopefully go through this journey together with some people, which would be nice. But, anyways, that's enough about that. So then you have cyclin-dependent kinases, which are really useful in mitosis and regulating the cell cycle, which we'll go over because mitosis is important. So is meiosis. Uh, I'm just envisioning that that's going to be a super long video, and I don't want to make it super long because if you're anything like me, your attention span probably isn't that long. just just for the record. So let me stay focused then. So cyclin-dependent kinases regulate the cell cycle. So I know you have the G1, G2 phases. I think there's another phase, the S phase, where you're like synthesizing uh, DNA. You're just like trying to get ready for the whole replication thing. So that's cyclin-dependent kinases. And then you can either activate or inhibit cell signaling pathways. Wow, pathways. Good God. Rush this. Pathways through phosphorylation. So then you have hydroxylation, which Red was involved in collagen. Hydroxylation is a hydroxyl group, which is an OH. Methylation. This decreases transcription because if you remember my earlier videos, I mentioned that there's methylation and there's acetylation. And we know that DNA is negative because it has a phosphate backbone. And phosphate we know is PO4. Um, it's 3 minus, PO4 3 minus. Yeah. Again, could be wrong, don't quote me, but PO4 3 minus and negatively charged phosphate backbone mixed with an acetyl group, which is negatively charged, is negatives are going to repulse. It's going to open up that DNA and not let it bind together super tightly around those histones. So that's going to increase transcription. Methylation is going to do the opposite. It's going to get rid of that negative charge, let those DNA negatively charged DNA or phosphate backbones bind to the positively charged histones and make it really tight. So you get a bunch of heterochromatin that's tightly condensed and decrease transcription. Acetylation would promote, euchromatin so that the genes can be transcribed. And then lastly, you have trimming. One example is pepsinogen. It's a protein or an enzyme that breaks down proteins in its pepsin form, but it has to be cleaved from pepsinogen to pepsin, and that's usually done by gastric acid. It's activated by gastric acid. So you eat a meal, stomach starts secreting hydrochloric acid or gastric acid um, from parietal cells. I'm pretty sure, um, and I know that that's all signaling by G cells, and you got gastrin, all this other stuff that we could do a video about that too, man. There's sciences. There's levels to this stuff, man. But I want to just give an example that pepsinogen can be activated by gastric acid and turns into it gets cleaved into pepsin, and that's the activated form which breaks down proteins. So yeah, like I said, quick video today. We just went over basically free ribosomes, endoplasmic reticulum, ribosomes on the endoplasmic reticulum, and post-translational modifications. This video was a follow-up to translation. I just didn't realize how long translation was going to take to explain. But here you go. Here's the entire notes, kind of, to review. There you go. I apologize. I wish I was better at drawing and more organized. I could probably improve on that. And then here we go. We know that peptides are released from the P site, whether they go into the lumen of the endoplasmic reticulum with the signal recognition particle, signal recognition sequence, and the signal recognition particle receptor. Open up those gates. I think I heard that those gates are called the translocons, but I'm not too sure. Don't quote me on it. I need to do more research so I can stop saying don't quote me on it, but until I know for sure. And then there you go, post-translational modifications. If you found this video even remotely useful, please leave a like, subscribe. I'm gonna keep on making these videos so we can learn together. Yeah, and it's just something to do, make studying a little bit more fun. Till next time, if you enjoy.